Welcome to Med in 5. The goal of our channel is to simplify medical concepts and help you understand them with zero fuss. Today, we're diving into a unique set of chlamydia-like bacteria, as they must live inside our cells to survive. Some questions to ponder: which organisms in this group form morulae inside immune cells? What are two preferred diagnostic tools for identifying obligate intracellular bacteria? What's the first-line treatment for host-dependent obligate intracellular infections in this group? Don't go anywhere. Obligate intracellular bacteria can't grow outside host cells, making them difficult to culture in the lab. They rely on the host's machinery to replicate. In some, peripheral blood smears show morulae or clusters of bacteria inside immune cells visible under the microscope. Diagnosis often depends on polymerase chain reaction and serology, which are rapid, sensitive, and specific. They're also considered atypical pathogens. They don't stain well with gram stain, and they don't respond to typical cell wall targeting antibiotics like beta-lactams. Doxycycline is the go-to treatment for most of them. Let's break them down. First is Ehrlichia chaffeensis, which causes Ehrlichiosis, a disease that targets monocytes. It's most common in the southeastern and south-central US, especially during warmer months when ticks are active. The lone star tick is the chief vector. Symptoms typically appear a week or two after a tick bite and include headache, fever, muscle aches, and low white blood cell counts. Some people also get a maculopapular or petechial rash, but that's less common. Older adults and immunocompromised patients are more at risk for severe disease. Blood smears may reveal morulae inside monocytes, but serology and PCR confirm the diagnosis. Doxycycline is the treatment of choice, even in children. Next is Anaplasma phagocytophilum, which causes Anaplasmosis, another tick-borne illness, but it infects granulocytes, especially neutrophils. Like Ehrlichiosis, Anaplasmosis presents with fever, chills, muscle aches, and low blood counts, but rarely a rash. The condition is most common in the northeastern and upper Midwestern U.S. The same tick that spreads Lyme disease, Ixodes scapularis, also spreads Anaplasma. Co-infection with Lyme disease is possible. Granulocytes may show morulae, but PCR and serology confirm the diagnosis. Again, doxycycline is first-line therapy. Let's move on to the Rickettsia genus, composed of obligate intracellular bacteria transmitted by arthropods. Rickettsia typhi causes murine typhus, aka endemic typhus. It's spread by fleas, especially in areas with rodent infestations, poor sanitation, or homelessness. Think southern Texas, California, and parts of Southeast Asia. At-risk patients include people in crowded urban areas or exposed to rodents. After a flea bite, patients develop fever, headache, and sometimes a faint rash that starts on the trunk. It's usually milder than epidemic typhus, but can still lead to complications like hepatitis or meningoencephalitis in severe cases. Rickettsia rickettsii causes Rocky Mountain spotted fever, one of the more severe rickettsial diseases. It's transmitted by dermacenter ticks, with cases mostly in the southeastern U.S. Campers, hikers, and outdoor workers are especially at risk. RMSF presents with high fever, headache, and a petechial rash that starts on the wrists and ankles, spreading to the palms, soles, and trunk. Rickettsia prowazekii causes epidemic typhus, spread by body lice in overcrowded, unhygienic settings like refugee camps. Symptoms include fever, confusion, and a widespread rash sparing the palms and soles. Rickettsia bugs don't produce morulae, but are usually diagnosed serologically. PCR helps when serology is inconclusive. Doxycycline is the drug of choice. Next is Coxiella burnetii, the culprit behind Q fever. Unlike the others, it's not spread by ticks or fleas. It's aerosolized, meaning people get infected by inhaling contaminated dust or particles, especially around livestock. Farmers, slaughterhouse workers, and veterinarians are at risk. C. burnetii causes atypical pneumonia without a rash. Patients may experience high fever, headache, and sometimes hepatosplenomegaly. Endocarditis may arise from chronic infection, especially in those with pre-existing valve disease. Q fever is diagnosed using PCR or serology. Doxycycline is again the preferred treatment. Let's review our questions. Which organisms in this group form morulae inside immune cells? Answer: Ehrlichia chaffeensis and Anaplasma phagocytophilum. What are two preferred diagnostic tools for identifying obligate intracellular bacteria? Answer: PCR and serology. What's the first-line treatment for host-dependent, obligate intracellular infections in this group? Answer: Doxycycline. Solved every question? Terrific! Like, share, and subscribe if you found this clip helpful. Download the summary table linked in the description. Thanks for watching.